Hello and welcome to BioCode. In this video, we will see the comparison operator within sequence record objects. For example, consider that you have multiple sequences available to you and you would like to test if one of them you would like to understand, you would like to find out if any of them are duplicates, redundant, or they actually match to each other. You can utilize any property of the sequence record. It can be an id, it can be name, it can be description, it can be annotations, it can be features, it can be letter annotations, but they should be together. For example, if you are trying to compare two sequence records, either you can compare their sequences together, either you can compare their ids or names, description, letters, or the annotations. You cannot compare annotation against id, you cannot compare id against sequence, you cannot compare one's id against one's name. That is not possible. So you need to understand that, and I will show you how it is useful for you. So first of all, we are actually trying to create two random sequences, and let's consider it is a different sequence. So we are actually trying to demonstrate that these are two sequence records, and one has ACGT sequence and the other has XYZ, which is not possible in the nature, by the way. But both of them have the same ID, which is test. Let's make it alphanumeric as well. Let's make it alphanumeric for the sake of simplicity, and so that we are on the same bioinformatics page. Okay, so now we understand that okay, our results, our IDs are same, but the sequences are itself themselves different. So we need to understand if both of these records are same. We do not care about the sequence, but we just need to understand if either of these sequences have their IDs to be the same. Let's run our code and then you will be able to see the results. And as you can see, there is a output as true. So that means both of their IDs are same. Let's run our code and let's compare the sequence record against sequence description. ID of one and the description of the other. Right? So let's run our code and see if it is possible. It is not possible because it is representing as false. Let's remove the property and let's run our code now. Then you will be able to see the sequence record comparison indelibility not implemented, and we need to explicitly define the attributes or the properties of a particular sequence record that we need to compare. So let's make it ID again. Okay, so how is it useful for you? What is the question behind it? What is the necessity of utilizing this sort of code in bioinformatics analysis? Suppose that you are working on multiple sequences and I'm sorry, sequence 1, sequence 2, sequence 3. Suppose you are working on these three sequences and all of them do have various different IDs, various different sequences. So you just need to comprehend over here, and all of them have different IDs, different sequences. They belong to the same species, however. So what I'm going to do is I'm going to utilize the ID of the first record, which is ABC123, and I'm going to run it through my directory where my files are present. I'm running a for loop, then I'm listing the directory, then I'm checking if the files